Hello, my name is Didier Stene. I'm a director at the Max Planck Institute for Heart and Lung Research in Bad Nauheim, Germany. And today we're going to be talking about cardiac trabeculation, which is a means by which the heart increases in size, mainly it increases its muscle mass, thereby allowing the growth of the organism. And we're going to be talking about this process in the zebrafish model system, which is the model system that allows live imaging, and is also fairly amenable genetically. And just to give you some introduction about the zebrafish heart, it starts beating at 24 hours post-fertilization. Here we're looking at 62 hours post-fertilization, and one can recognize a number of features. The chamber right here is called the atrium, and it's the collecting chamber. And then the blood moves into the ventricle, which is the pumping chamber. And here we're labeling just the myocardium, or cardiomyocytes, so the muscle cells of the heart. And we're using a microscopy technique called selective plane illumination microscopy, or SPIM, which was revived right here at EMBL. And Jan Heusken, a former postdoc in the lab, was instrumental in this technology. We can label multiple cells, of course, and here we're looking in dark red the cardiomyocytes, the muscle cells. In green, we're looking at the endocardial cells, or endothelial lining cells in the heart. And then in bright red, the blood cells. And again, we're looking at SPIM imaging, so looking at single plane images. And this is now at 5 DPF, 5 days post-fertilization. And again, here we're looking at the atrium and the ventricle. And you can see these right here. These are the so-called trabeculae. These are multicellular protrusions that form in the lumen of the ventricle. And you can see, and we'll come back to this point later on, but this is now the inner curvature, and this is out here is the outer curvature. And these trabeculae are formed in the outer curvature, but not in the inner curvature, and not in the atrium. Now you can see also, and we'll come back to this later on, the different blood flow patterns in the ventricle in terms of the inner curvature versus the outer curvature. So one can also, of course, look at these organs in a 3 dimension with time, so in 4 dimensions. So imagine you're sitting inside the ventricle looking out, and this is what you'd be looking at. So these are muscular structures right here, and these are what we were looking at essentially in cross-section earlier on. So these are these muscular ridges, the trabeculae. And so essentially taking these cartoons from the mouse heart and just looking again at the ventricles right here. And you can see in these ventricles, these ridges, which are the trabeculae, both in the left and the right ventricle. Looking at this at high magnification, one can recognize the compact layer cardiomyocytes from the trabecular cardiomyocytes. And then right adjacent to that, right here in blue, are the endocardial cells. And very similarly, in this zebrafish, one can distinguish between the compact layer cardiomyocyte, so a very thin monolayer of cells, and these ridges, which we were looking at earlier, which are called the trabeculae. So essentially, as I mentioned, there are a number of interesting patterning questions or developmental questions about the formation of trabeculae. And just looking at this cartoon, as I mentioned, they're in the atrium, the collecting chamber, there are no trabeculae. But in the ventricle, these trabeculae are formed in the outer curvature, but not in the inner curvature. And when we look back at the movie in a few minutes, you'll see again these different flow patterns. And it's more of a perturbed flow in the outer curvature, more of a laminar flow in the inner curvature. And this already suggests that flow may in fact be important for the formation of these trabecular structures. So this is, I'll just show you a few movies of trabeculation over time. We'll just look at 3 different time points. Trabeculae start forming at around 60 hours post-fertilization, usually directly adjacent from the atrioventricular canal. And again, you recognize not only the trabeculae right here, but I also want to point out your attention to in the, essentially in the atrium, the separation of the space between the red and green cells, between the myocardial and endocardial cells, is filled by a specialized extracellular matrix called the cardiac jelly. And you can see this more clearly again at 5 days, this movie I showed you earlier, looking essentially again here at the atrium. You can see that the space between the green and red cells is fairly substantial at this stage, especially in the atrium, but not in the ventricle itself. So the later stage that we can look at easily using this microscopy technique is 14 days. And again, you can see the compact layer cardiomyocyte right here, very thin layer. And most of the myocardial mass is in fact in the trabecular layer. So not only most of the cardiomyocytes, most of the muscle cells in the trabecular layer, but now if you look at them at the ultrastructure, specifically looking at sarcomeres, using now a line which labels actin filaments, you can see that these trabecular layer cardiomyocytes look very different from the compact layer cardiomyocytes. You can see in the compact layer cardiomyocytes, these actin filaments are loose, they form a loose mesh of filaments, whereas you can see these pronounced sarcomeres in the trabecular layer. And in the next movie, I'll show you, in fact, again, this is a SPIM movie focusing on the single plane, and of course, as the heart contracts, you'll see the trabecular layer and compact layer cardiomyocytes coming into focus, so you can appreciate the difference in the actin filament distribution in these two different cell types. So not only are mostly the cardiomyocytes in the trabecular layer, but the ones that are doing most of the work, because they have more pronounced sarcomeres, are also the trabecular layer cardiomyocytes. So they're of course very important for the function of the heart as it pumps blood throughout the organism. And so again, imagine you're sitting inside the ventricle looking out, and here in yellow we've outlined or essentially painted these trabecular layer cardiomyocytes. Here in red we have essentially then labeled the nuclei of these cardiomyocytes, and I'll just rotate this so you can see how these cardiomyocytes, these trabecular cardiomyocytes, are in fact filling the entire chamber. Looking at the shape of these cardiomyocytes by doing cell transplantations at early stages, early embryonic stages, you can see, for example, that the trabecular layer cardiomyocytes shown right here present a forked appearance, whereas the compact layer cardiomyocytes present a more smooth appearance. And again, this allows these trabecular cardiomyocytes to form these multicellular muscular ridges that essentially bring the wall of the ventricle together as they contract. So let's think about the cell biology underlying this trabeculation process, specifically how essentially a compact layer cardiomyocyte, as you can see here, essentially would move out towards the lumen and start seeding this trabecular layer. And so one can imagine a number of scenarios, including the two shown here. So one would be a simple delamination way by which this red cardiomyocyte, which essentially migrate away from the compact layer and start seeding the trabecular layer. Another process might be oriented cell division, whereby, again, this red cardiomyocyte would divide. One of its daughters would essentially seed the trabecular layer, and the other cardiomyocyte would stay in the compact layer. So we carried out a number of experiments, including making small clones of labeled cells, or labeling small clones of cells. We also did some sophisticated lineage tracing using the Cree-ER technology. We looked at the proliferation of these cardiomyocytes at this stage. In the fish embryo, there's very little proliferation of cardiomyocytes, in fact. And all the data we generated, in fact, pointed to the first model, the delamination model, being mostly at play. And so thinking about this delamination process and thinking about essentially how these cardiomyocytes may be oriented in the apical-basal orientation, we labeled one cardiomyocyte using a basal marker that came from a C. elegans forward genetic screen called PAR1. And so you can recognize that this red marker now lies closest to the lumen. But of course, it's separated from the lumen by a layer of endocardial cells and also a layer of extracellular matrix, the so-called cardiac jelly. So essentially, on the abluminal side, so away from the lumen, we have the apical side, and then on the luminal side of the cardiomyocytes, we have the basal side. And so if you think about this process of delamination, it's possible that it's driven by a process called apical constriction, whereby the apical surface or abluminal surface of the cardiomyocyte would constrict, and this would allow or drive the delamination of the cardiomyocytes. And so David Stout, a student in the lab, an MSTP student in the lab, decided to look for apical constriction, again using live imaging. And after spending a number of years developing a processing software for live and using this software for imaging and image reconstruction, he was able to generate movies such as this, where he was able to, in fact, find evidence for apical constriction. But in addition, he found evidence for the formation of protrusions, and these turned out to be basal protrusions. So again, looking and focusing on the red arrow here, this is a single cardiomyocyte right here, and you'll see that in this overnight movie, this cardiomyocyte is sending out the basal protrusions. And about 50% of the cardiomyocytes are in fact sending these protrusions. So looking at this process in cross-section and labeling here a few cardiomyocytes, you can see in purple right here, again, we're looking now at a surface view, so this is the abluminal surface of these 4 cardiomyocytes. In purple is the one that's shrinking its abluminal surface. And so again, you can see over time that this surface, this abluminal or apical surface of the cardiomyocyte is becoming smaller. And if you look at cross-section, you can see, in fact, that this purple cardiomyocyte starts in the compact layer, shrinks its apical surface, sends out basal protrusions, and then eventually it delaminates completely, loses contact with the abluminal surface of the heart, and then is then ready to start sealing the trabecular layer. So essentially what we have then is a process of delamination driven by basal protrusions in apical constriction, and so we're ready then to start looking for factors regulating this process. And we initially took a forward genetic screen, so we conducted a pilot screen, and essentially we identified a number of mutations where trabeculation was defective, but it turned out that all of these mutations also affected the function of the heart, the contractility of the heart. And as we saw in the first part, when you affect the contractility of the heart, you also affect its maturation, not only the formation of the valves, but also the formation of trabeculae. And so this was not a productive way to identify genes that regulate the formation of trabeculae. So we went back and looked more carefully at the mouse literature, and when mutations were initially first made in the mouse, two of the genes that were initially mutated are shown here. This is neuregulin, a ligand, for the receptor ERBB2. And you can see again the wild-type hearts here, these ventricles that I showed you before with the pronounced trabeculae, and in the mutant, both in terms of the ERBB2 receptor as well as the ligand neuregulin, you can see in fact that these trabeculae are missing. And so this led to a model backed up by additional data that essentially suggested that neuregulin, which is expressed, as you can see here, by the endocardial cells, will bind its receptor ERBB2, which is expressed by the cardiomyocytes, and this will lead to a series of events that leads to the formation of these trabeculae. And so we wanted then to use the zebrafish to see if we could get additional insights into this process. And fortunately, we're able to find an ERBB2 mutant from Will Talbot at Stanford, and this mutant had come from a screen for myelination mutants. And you can see again, on the left, they are the wild type, and on the right, the mutants. And you can see that both at early and late stages, these mutant hearts are completely lacking trabeculae. So very much like the mouse phenotype, or as shown by the mouse phenotype, ERBB2 zebrafish mutants completely lack trabeculae. One of the questions we wanted to ask is what would happen if you took a mutant cell and put it into a wild-type heart? Would that mutant cell now essentially go along for the ride and be able to populate trabeculae, or would it always stay in the compact layer? And the way you address this question in zebrafish, the way you make genetic mosaics, is by cell transplantation at an early embryonic stage, as shown here. And so essentially you take fish that are transgenic and expressing different color transgenes, fluorescent transgenes in their cardiomyocytes. You take a few donor cells right here, and you put them into the host, and essentially then you let the embryo develop and look at its heart. And in a number of cases, then you have contribution of the donor cells into the host heart. And so, for example, here, this is a control experiment where we've taken red cells and put them into a so-called green heart. And you can see that these red cardiomyocytes now are populating both the trabecular layer as well as the compact layer. If you do the experiment now and start by taking mutant cells right here in red and put them into a wild-type heart in green, you can see that all the red cells are in fact in the compact layer. There are no red cells in the trabecular layer. Conversely, if you now take wild-type cells in green and put them into a mutant heart in red, you can see that the only cells in the trabecular layer are in fact green. There are no red cells in this trabecular layer. So there's a very strict requirement, cell autonomous requirement for ERBB2 in the formation of trabeculae. And so essentially, this brings me to the first summary, and I've shown you that at the cell biological level, these cardiomyocytes appear to form trabeculae by delamination, and this delamination process appears to involve basal protrusions and apical constriction, and it requires ERBB2 signaling. I mentioned the role of flow before because, in fact, this trabeculae only appear in the outer curvature, not in the inner curvature, and flow patterns are very different in these two regions of the heart. It turns out that if you block flow or contractility, in fact, prevent the formation of these trabeculae. And we'll come back to this point later on, trying to understand how, in fact, flow and contractility regulates cardiac trabeculation. So these further experiments have taught us a number of things. One is that ERBB2 signaling regulates actin dynamics, and essentially it involves, actin dynamics is involved in a number of processes, including the formation and stabilization of these basal protrusions, and ERBB2 signaling is required for this actin dynamics. From work in a number of model systems, including looking at breast cancer, ERBB2 has also been implicated in regulating epithelial to mesenchymal transition. And so one of the questions we'll address in the next few minutes is whether, in fact, there is also an epithelial to mesenchymal transition in this process of trabeculation. And clearly, ERBB2 signaling is going to be involved in regulating many other processes, and we're now busy trying to identify, using a phosphoproteomic approach, identify some of the downstream pathway of ERBB2 signaling during the process of cardiac trabeculation. Data in a number of other model systems, including chick and mouse, have implicated a number of other signaling pathways in cardiac trabeculation, and these signaling pathways include Notch, BMP, and nephrin, and we're now also looking at some of these pathways using some of the advantages that I mentioned, including the live imaging and the ability to get single-cell resolution of this process in this beating heart. So I want to finish off with asking three questions. As I mentioned, one major issue is this apical basal polarity and this question of whether epithelial mesenchymal transition is in fact taking place during the delamination process. We'll look at the neuregulin ligand, and by looking at this question, we'll be able to address whether, or in fact why, trabeculae occur only in the ventricle and not in the atrium, and also what the role is for cardiac function, that is flow and contractility, in the formation of this trabeculae. And then finally, I'll just show you a few slides about one of the challenges that these cardiomyocytes face as they delaminate and seed this inner layer, this trabecular layer. So let's start with the first one, this question of apical basal polarity. So the working hypothesis is very simple and essentially states that before trabeculation starts, these cardiomyocytes are in fact polarized in the apical basal axis. So the proteins present on the apical surface or abdominal surface would be different from the proteins present on the basal surface. And the second hypothesis would state that then ERBB2 signaling is in fact regulating the loss of this apical basal polarity as the cells start to delaminate. And so to address this hypothesis, what Vanessa did is generated a transgenic line that labels specifically the apical membrane of these cardiomyocytes, here shown in green, and another transgenic line that specifically labels the red membrane shown here, the basal side. And I'll just focus on the apical marker, which is shown right here. And so let's just focus, so this is now prior trabeculation right here, and this is after trabeculation. And here in red we're labeling essentially the entire membrane of cardiomyocytes, so all around the cortex. And here in green is this apical marker, and we're using a podocalixin protein, which is the apical protein. And you can see very clearly that this green marker now is labeling only one surface of the cardiomyocytes, and that's the abluminal surface. And this would be, for example, just right here, would be a single cardiomyocyte. This is prior to trabeculation, and then after trabeculation, you can see now that this apical marker has become relocalized all around the cortex of cardiomyocytes that have left the compact layer and have now entered the trabecular layer. So we can use this line to also do some live imaging, and here I'm just showing you a few snapshots before I show you the movie. But again, just focus on the arrow right here, and of course, as this abluminal surface is shrinking, you have, of course, consolidation of the GFP, the podocalixin GFP protein on this shrinking surface, and that's why, of course, it becomes more prominent, more visible in the confocal microscope. So just focus on the arrow again, and in this movie, and you can see the accumulation of this apical marker on the shrinking part of this cardiomyocyte that eventually then will leave this compact layer and enter the trabecular layer. Now we can use this apical marker, as shown here, to quantify the number of cardiomyocytes in the compact layer that are undergoing this apical constriction. And we can look this, using this method, we can see in fact what the role of ERBB2 signaling as well as the role of flow is in the formation or the appearance of these apical constrictions. And what we see when we block ERBB2 signaling, in this case we're using a small molecule inhibitor to block this receptor tyrosine kinase, and you can see that blocking ERBB2 signaling leads to the loss of the appearance of this apically constricting cardiomyocytes. And similarly, if you block flow, in this case we're blocking contractility, using a myosin TPase inhibitor using a small molecule called BDM, you can again lose the appearance of these apical constrictions. So essentially what I've told you is that in fact, yes, these cardiomyocytes are polarized prior to trabeculation, that the abluminal surface is where the apical surface of the cardiomyocytes is, and the other surface, the basal surface, is in fact the luminal surface, and this is the same in the mouse as we've shown also. Now we can visualize live and quantify this apical constriction as the cells depolarize and delaminate. So essentially the cells lose this apical basal polarity as they start delaminating. And we've also seen that in fact, and I won't have time to show you the data, but that Notch signaling is activated in the cardiomyocytes that are directly adjacent to those undergoing apical constriction. And so one interesting question about trabeculation is what selects the cardiomyocytes that in fact undergo this process, this behavior. As I mentioned, about 50% of the cardiomyocytes undergo or exhibit these basal protrusions, but only a subset of them will in fact delaminate and start seeding the trabecular layer. And so it's been a question in the field as to what selects the cardiomyocytes that go into the trabecular layer versus those that remain in the compact layer. And then finally, and because Notch is activated in these cardiomyocytes that stay in the compact layer, we wanted to test whether in fact there was a role for Notch in this pathway, but in our hands, Notch signaling, blocking of Notch signaling does not appear to increase the number of cardiomyocytes undergoing apical constriction or those ending up in the trabecular layer. So this brings me to the second point that I want to talk about, which is the ligand issue. And in the mouse, neuregulin-1 has been shown to be the ligand that binds ERBB2 and drives cardiac trabeculation. So in the fish, we started by looking at neuregulin-1, and we made mutations in this gene, but in fact, as you can see here, we're looking at the wild type on the left and mutant on the right, and you can see that there are no differences between the mutant and the wild type. So neuregulin 1 is in fact not required for cardiac trabeculation in zebrafish. We turned our attention to the other neuregulin genes, and specifically to neuregulin 2A. And the reason we looked more specifically at this gene is because, much like the ERBB2 mutants, these mutants die between 8 and 12 days post-fertilization. We're using an allele from Steve Ecker and Matthias Hammerschmidt that's in fact a protein trap, and so this allows us to look at the expression of this gene by looking at, get a sense of the expression of the gene by looking at the expression of the protein, here shown in red. And you can see that, again, compared to the wild type on the left, you can see that the mutants, the neuregulin 2A mutants, are in fact lacking trabeculae. Here in red is the expression of this protein trap, so neuregulin 2A. And so one can look at essentially higher magnification. You can see, again, just focusing on these arrows right here, the yellow arrows are cells that are expressing neuregulin 2A. These are endocardial cells, and they're directly adjacent to these forming trabeculae. So these endocardial cells that express neuregulin 2A, we are of course expecting to see them in the ventricle, which is where trabeculae occur. But surprisingly, we also saw evidence for neuregulin 2 expression in the atrium. And so the question remained then, why are there no trabeculae in the atrium? And so to address this question, we took a slightly different approach. And that is that we expressed a neuregulin 2A specifically in cardiomyocytes. So this is now ectopic expression. As I mentioned, neuregulin 2A is normally expressed in the endocardial cells, not the cardiomyocytes, not the myocardial cells. So we took this transgenic approach, and so essentially this is just the way the construct was designed. And so we're using DT tomato here to label the cells that are expressing neuregulin 2A. And essentially what you see is as you ectopically express neuregulin 2A in cardiomyocytes, that you can not only rescue the mutant, the neuregulin 2A mutant, but you're driving essentially a trabeculation-like behavior in the chambers, both in the ventricle as well as in the atrium, as you'll see much better in this cartoon. So again, on the left is essentially the control heart, no transgene expressed. But when you express now ectopically neuregulin 2A in the cardiomyocytes, you can see both this trabeculation-like behavior in the ventricle, but also right here in the atrium. Essentially, these data tell us that atrial cardiomyocytes are competent to respond to neuregulin 2A. So the other question I wanted to address using this tool is why cardiac contractility and flow are in fact required for correct trabeculation. And so these are some of the data initially generated. So if you now block cardiac contraction using, for example, a mutation in cardiac troponin T, or use this ATPase inhibitor I mentioned earlier, BDM, you block contraction, and as you see, you also block the formation of trabeculae. And it turns out that the reason this occurs is because now if you look at the expression pattern of neuregulin 2A, again using this protein trap allele I mentioned earlier, you can see again the wild type here on top and the mutant at the bottom. So in this case, we're using a morpholino against cardiac troponin T to block contraction. And you can see that you're losing most of the expression of neuregulin 2A, especially in the ventricle. And as you lose neuregulin 2A, much like you see in the neuregulin 2A mutants, you essentially fail to form trabeculae. So here we're expressing neuregulin 2A, specifically in non-contractile cardiomyocytes. As you can see, we're using cardiac troponin T morpholinos to silence the heart. And then we're overexpressing, as you can see by this red fluorescence, overexpressing neuregulin 2A in the cardiomyocytes. And this leads to a trabeculation-like behavior, again, indicating that these cardiomyocytes, despite being non-contractile, are competent to respond to neuregulin 2A. So in summary, what I've shown you is that in terms of the ligand driving trabeculation behavior in the zebrafish, unlike in mammals, it's neuregulin 2A, not neuregulin 1, which brings in interesting evolutionary issues. That neurogranin 2A is expressed in all endocardial cells, including those in the atrium. Of course, there may be differences in the level of expression, but this protein trap is not the ideal reagent to look at this. And also that neurogranin 2 expression is regulated positively by flow cardiac contractility. That is, if you lack flow or cardiac contractility, you lose neurogranin 2 expression. And then finally, that the cardiomyocytes in the atrium, as well as the non-contractile cardiomyocytes, are in fact competent to respond to neurogranin 2A signaling. So now I'll finish off with the last point I want to make here, which is the challenge that these cardiomyocytes are facing as they delaminate from the compact layer. And of course, they need to remain attached to their neighbors as they do this in order to form essentially a fully functioning organ. And so we started looking at cadherin, and cadherin specifically, which is a major adhesion protein in cardiomyocytes. And as you can see here, so this would be now a single cardiomyocyte right here. And on the lateral sides of these cardiomyocytes, and just to remind you, these are not epithelial or epithelial-like cells with a clear apical-basal polarity. And you have N-cadherin expression specifically on the lateral sides. And then as you move a few hours later, you can see that there's redistribution of this N-cadherin protein. And this is better seen if you just look at the surface view. Again, this would be now a single cardiomyocyte in the inset here on the left. And you can see, of course, then the lateral distribution of this N-cadherin GFP protein. And now, as you move a few hours later, you can see redistribution so that it's not just on the lateral sides, but also has come now either to the apical or the basal side. And you can distinguish this by looking at the cross-section. And so very clearly what you see, and again, this would be a single cardiomyocyte in the compact wall, a single cardiomyocyte in the trabecular layer, and that you have redistribution of N-cadherin now to the basal side of compact layer cardiomyocytes. And of course, so just shows in the cartoon fashion, we start with N-cadherin on the lateral side of cardiomyocytes, and then redistribution to the basal side. The trabeculating cardiomyocytes now have N-cadherin all around their surface, and this allows then the adhesion between the trabecular cardiomyocytes and the compact layer cardiomyocytes. And so again, just to summarize, what I've shown you is that this process of cardiac trabeculation, which is required to increase the muscle mass of the heart before the formation of the coronary vessels, requires ERBB2 signaling. It also requires flow and contractility, and the way flow and contractility are required is by regulating the expression of the ligand, neurogranin 2A, in the case of the zebrafish. It's the ligand for ERBB2. That we have in terms of the cell biology of the process, we have a clear apical-basal polarity of these cardiomyocytes prior to delamination, and there's an appearance of basal protrusions as well as apical constriction to drive the delamination process. And I won't have time to go into it, but essentially the endocardial cells are not only secreting neurogranin 2A and activating the ERBB2 receptor, but they may also play a more active, a more physical role in the trabeculation process, possibly by making stable junctions, stable contacts with the cardiomyocytes that are in fact delaminating from the compact layer. And well, I want to acknowledge many past and present members of the lab who have been contributing very significantly to these projects, as well as the funding agencies, both initially at UCSF and now in Germany. Thank you.